shunt. Other names are pentose phosphate pathway or phosphogluconate pathway. The importance of HMP shunt is mainly due to NADPI2 produced in this pathway. NADPI2 is the major and the most important biochemical reductant. There is no exact definition of the pathway because it produces many sugar intermediates for different carbohydrate metabolic processes, and unlike glycolysis it is multi directional. It consists of two irreversible oxidative reactions followed by series of sugar phosphate interconversions. It occurs in the cytosol of erythrocytes, livers, adipose tissues, lactating mammary glands, thyroid, adrenal gland, cortex, testes, and ovaries. Some important points about the steps of HMP shunt. No need to remember the steps. Glucose to glucose 6 phosphate is uh, not the first reaction, neither it is a part of HMP shunt. Rather, it is a common reaction for both glycolysis and HMP shunt. HMP pathway starts with glucose 6 phosphate. HMP shunt is also called direct oxidative pathway because oxidation occurs in the first reaction, glucose 6 phosphate to glucose 6 phosphogluconolactone with the productions of one NADPI2 of the product. The second NADPI2 is produced by oxidative carboxylation of 6 phosphogluconolactone with the release of carbon dioxide. This carbon dioxide is the characteristic product of HMP pathway. Rate limiting enzymes. Glucose 6 phosphate dehydrogenase is the key enzyme. The enzyme produces one NADPI2 of the pathway. Regulation. The enzyme glucose 6 phosphate dehydrogenase is regulated by NADP over NADPI2 ratio. Increase NADP over NADPI2 ratio stimulate the pathway to produce more NADPI2. Decreased NADP over NADPI2 ratio inhibits the pathway. Importance of HMP shunt. In this pathway, not a single ATP is directly consumed or produced. It is the only source by which body gets reduced form of NADP, for example, NADPH2. Two molecules of NADPH2 are produced for each molecule of glucose 6 phosphate entering the oxidative part of the pathway. Pathway maintains the integrity of RBC by producing NADPH2. Pathway produces pentoses ribose which are required for the biosynthesis of nucleotides, RNA and DNA. It produces immediate products for glycolysis. Uses of NADPI2. NADPI2 is a biochemical reductant and is the reduced form of NADP. Coenzyme NADP is a derivative of niacin, water-soluble vitamins. NADPI2 helps in the biosynthesis of fatty acids and cholesterol. It causes reduction of hydrogen peroxide and nucleic superoxides. Hydrogen peroxide and superoxides are oxygen derived free radicals which are highly reactive and can cause serious damage to RBC, DNA, proteins, and unsaturated fatty acids. It reduces glutathione of RBC. Glutathione is a tripeptide present on the cell membrane of RBC, which on oxidation makes the RBC fragile. So, in order to survive and maintain the integrity of RBC, it is very important to keep the glutathione in a reduced form. NADPI2 is required by the liver microsoma cytochrome P450 monooxygenase system, cytochrome P450 system for the hydroxylation of steroid, alcohol, and many drugs. NADPI2 is also required by WBC for phagocytosis and killing of bacteria. Comparison between glycolysis and HMP shunt. Clinical notes. Glucose 6 phosphate dehydrogenase deficiency. It is an inherited X-linked disease, therefore only males are affected and females are only carrier. It is very rare for female to have glucose 6-phosphate dehydrogenase deficiency. It is the most common disease producing enzyme deficiency. Deficiency of glucose 6-phosphate dehydrogenase results in deficiency of NADPH2. Deficiency of NADPH2 increases the concentration of oxidized glutathione. Oxidized glutathione is unable to keep the cell membrane of RBC in reduced form, hence RBC becomes fragile. RBC become weak and vulnerable to oxidative damage by hydrogen peroxide, superoxides, and oxygen-derived free radicals, resulting in hemolysis. Glucose 6 phosphate dehydrogenase deficiency is characterized by hemolytic anemia only when a person is exposed to oxidizing agents, hydrogen peroxide and superoxide. Interestingly, people with glucose 6 phosphate dehydrogenase deficiencies are resistant against falciparum malaria. Precipitating factors, oxidant drugs, 3 A's, antibiotics, anti-malaria, and antipyretics. Ingestion of our beans in Mediterranean regions, severe infections, inflammatory response generates free radicals which causes oxidative damage. Signs and symptoms: neonatal jaundice, chronic hemolytic anemia. Patients do not show clinical manifestations unless and until they are exposed to the above mentioned precipitating factors. Investigation: evidence of hemolysis, laboratory investigation about absent or low level of glucose 6 phosphate dehydrogenase. Treatment: any offending drugs should be stopped. Underlying infection should be treated. Blood transfusion may be life saving. Splenectomy is usually not helpful. Cori's lactic acid cycle. It is also simply known as lactic acid cycle. It is a simple metabolic pathway in which lactate produced by anaerobic glycolysis in the muscles moves to the liver and is converted to glucose, which then returns to the muscles and is converted back to lactate. Importance: produces ATP for muscle activity through glycolysis in muscles, prevents lactic acidosis in the muscles under anaerobic conditions. Metformin, anti-diabetic drug, inhibits Cori cycles, precipitating lactic acidosis in patients with renal failure. Glycolysis. It is the breakdown of glucose up to pyruvate or lactate. It's transfer of glucose. There are two types, aerobic and anaerobic. ATPs are utilized as well as produced. It produces NADH2. It occurs in all cells of the body. Oxidation does not occur in the first step. Carbon dioxide is never produced. No pentoses are produced. Only one molecule of glucose is required for the complete process. The end product pyruvate is dependent on TC cycle for its complete oxidations. HMP short, it is the breakdown of glucose to yield different metabolic intermediate. The end products are two molecules of fructose 6 phosphate and one molecule of 3 phosphoglyceraldehyde. It's transfer from glucose 6 phosphate. There are no subtypes. No ATP is produced are utilized. It produces NADPH2. It occurs mostly in those tissues where lipids and steroid synthesis takes place. For example, adipose tissues, lactating mammary glands, adrenal cortex, uh, thyroid, testes and ovaries, liver, and RBCs. Oxidation occurs in the first step. Production of carbon dioxide is a characteristic features. It produces pentoses, riboses, and ribuloses. The three molecules of glucose are required for the complete oxidation. The end products are not dependent on TCS cycle. Thank you very much.